Hey everyone, welcome to this video. And as in last class, we have discussed about amino acids. Oh, wait a minute. As we discussed about the amino acids, so we discussed about the amino acid structure. So there's a alpha carbon to which a carboxylic acid group and an NH3 group is attached, and there is a hydrogen, and then there's a R group. Okay, and this R decides what kind of amino acid it may be, right? But one thing that I forgot in the last class, let it be the continuous part of this particular lecture, that this particular amino acid does not exist in NH3 or the COH form. Okay, it exists in the zwitterion form. Okay, and what is this zwitterion? So in body, the overall structure is something like this: C alpha, COO negative, then NH3 positive, R group, and H group. Okay, so this is the structure in our physiology that you must remember. So this is called zwitterion structure. Okay, and this R group has several roles in it. Okay, so we'll come to this part. So this R group has properties of these amino acids. So it dictates how and why a protein should fold, because this R group can be hydrophilic, it can be hydrophobic, it can be a charged species, or it can be neutral species altogether. Okay, neutral species altogether. So if we understand the basic like different amino acids, so we have a glycine, we have alanine, valine, leucine, isoleucine, methionine, phenylalanine, proline, serine, threonine, cysteine, asparagine, glutamine, tyrosine, tryptophan, aspartate, glutamate, histidine, lysine, and arginine. I know there are total 20 amino acids, and you have to learn every bit of this because sometimes question come directly from the structure. So they are going to give you di or tripeptide. For example, they are going to give a structure like this. So there is NH3CCO, there's a bond, and then there's another C. Let's suppose there is CH2, and then there is this particular thing, and again. So this kind of dipeptide. Now you have to identify this structure. Okay, so what is this dipeptide? So you have to give name, like which amino acids are there. Okay, now to understand that, so there's a division like I said. So there are hydrophobic amino acids, so which are alanine, valine, phenylalanine, tryptophan, leucine, isoleucine, methionine, and proline, and also glycine. Okay, and glycine is one of those amino acid which is not chiral because the R group here is hydrogen only. Okay, so if you keep the mirror image of the same, so they are going to overlap because of this hydrogen. So just remember that the glycine is not chiral in nature. So this might be asked during your IIT jam, especially I'm saying IIT jam preparation. Okay, also as you can see, so every colored species is the R group here. Okay, so for example, alanine has CH3 group. Okay, so you simply substitute R with CH3 and you're done. So this is alanine. Okay, so valine has this CH3. Okay, so valine is something like this. So this is C, CH3, and H. Okay, attached to the main alpha carbon. I hope you get it right. So there's a tryptophan. So there are 3 amino acids which are also called aromatic amino acid, which are tryptophan, which are not histidine, but where is it? Okay, phenylalanine and tyrosine. Where is tyrosine? I'm not seeing the tyrosine. Oh, there it is. And tyrosine. Okay, so these are 3 amino acids which are essential. You must know about these 3 amino acids because they help in quantifying the protein, and how it does that we are going to understand in next few classes. But understand this, I know this class might be boring, but you need to understand this because you have to understand the protein structure, and based on this protein structure we are going to answer very of the question in upcoming classes. Okay, so like I'm saying, so there are hydrophobic amino acids, so where the group is very hydrophobic and this does not dissolve in water. So like CH3, 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 phenylalanine, tryptophan has this CH, and then there is this group NH as well as the benzene group attached to it. I'm not sure what it is called. And then there is leucine, CH2, CH, CH3 and CH3. Okay, so if you have to remember all of this, okay, make sure you draw a diagram by yourself. Okay, so first you write NH2, C alpha, COH, H and R group. Okay, so this is your basic amino acid. Now what is alanine? Alanine has one CH3 group in it. Okay, so you simply replace this CH3. Okay, so that is NH2, C alpha, CH3, COH and H, right? What is glycine? We are simply going to remove this CH3. So for example, we'll simply remove this CH and we do the H replacement there. This becomes glycine, right? The only thing that we need to change is this H group and everything is going to be fine. Okay, so make sure you write each and every of this structure so that you remember everything, right? We have amino acids and we have R groups, right? And we have one letter abbreviation for each of this particular amino acids, and there is 3-letter abbreviation that you need to consider. Okay, for glycine we write G and then there is a 3-letter abbreviation as GLY. For alanine it is A and then ALA. So you have to remember each of these for your IIT exam, GAT-B gate preparation, because question comes directly from the dipeptide and tripeptide structure. So that's why you must know the single letter as well as the 3-letter abbreviation of the amino acids. Okay, now coming for the R group. Okay, so we have hydrophobic amino acids, we have polar amino acid, and there are charged amino acid, right? And then there is a neutral species, which is this, right? So for the hydrophobic part, so the R group that we are having is mostly carbon source, right? So there are directly carbon chain, right? So here we have alanine, valine, phenylalanine, tryptophan, proline, methionine, isoleucine and leucine, right? And of this, methionine is the only one which has sulfur in it. Of all the amino acids, there are only 2 amino acids which have sulfur in it. One is methionine and another one is cysteine. But methionine sulfur does not play any role in bond formation. Just remember that. Sometime question directly comes from it, that which amino acid sulfur is used for the bond formation, and it is always cysteine. Okay, so these all amino acids are hydrophobic in nature, and proline is another amino acid which is not actual amino acid, rather it is a amino acid because of this particular group that you are seeing here, right? Next come the polar amino acid. So polar must have OH or SH group in it. So whatever structure has OH and SH group in it will be the hydrophilic, as well as the NH2 positive or group, these will be hydrophilic, right? So for polar amino acid we have serine, we have threonine, again there is CH2OH, CH3CHOH, then there is benzyl OH, again there is in cysteine we have CH2SH, in asparagine we have CONH2, glutamine we have CH2CONH2, right? We have histidine, again there's a formation of NH, right? And then the glycine is not hydrophilic. Okay, so I hope you get it. If you do not get it, do not get me confused. Nobody can teach you these much better. Only thing that you need to do is you need to make these diagram by yourself, and then only you'll be able to master it. Okay, for the aspartate we have CH2COOH group, right? So this particular H, when is released in the system, so there is a formation of this negative, due to which overall charge in amino acid becomes negative. So we have aspartate, which is negatively charged amino acid, and then we have glutamate, which is again negatively charged amino acid because there is the addition of 1 carbon more, right? CH2, and then there is CO negative for the R group, right? And then we have lysine. So lysine has 1, 2, 3, 4 carbon, and then there is the formation of this NH3 positive group, right? For the arginine, there is the same thing. So there is CH2, CH2, CH2. So 3 carbon. Then there is NH, and then there is another carbon. So total 4 carbon and 3 NH group. Okay, just remember that there is 3 NH group that are present there. So thus we have arginine in the system as positively charged amino acids. So both the lysine as well as arginine are the positively charged amino acids. Please do it by yourself, make the diagram by yourself, and you'll see the pattern that which amino acids are correlating and which amino acids are not correlating, and based on that we are going to see the difference. And in next class we are going to learn more about these things.